Chapter 8 Feet. Introduction. Microsporidia are classified under phylum Microspora. They minute, intracellular, gram-positive, spore-forming protozoa. Microsporidia are also classified based on their habitat and the infections caused by them. Table 1. History and distribution. Microsporidia are of historical interest as they the first protozoan parasite to have been successfully studied and controlled by Louis Pasteur in 1863. During an investigation of silkworm disease epidemic in France, it was this experience which led Pasteur to his epochal work on human and animal diseases that formed the foundation of microbiology. The Table 1. Classification of Microsporidia species. Habitat and infection. Causative agent of the silkworm disease, pebrine, is Nosema bombycis, a microsporidian parasite. Microsporidia had been known as animal parasite for long, but their role as human pathogens was recognized only in the mid-1980s with the spreading of acquired immunodeficiency syndrome, ALDS. Some 9 genera and 13 species are associated with human disease, particularly in the human immunodeficiency virus, HIV, infected and other immunocompromised subjects. Morphology. Microsporidia are unicellular, obligate intracellular parasite. They reproduce in host cells by producing spores. Sporogony. Genus Enterocitozoon e benusi, small intestine epithelium, leading to diarrhea and wasting. Also found in biliary tract of patients with cholecystitis. Rarely spreads to respiratory epithelium. Encephalitozoon e intestinalis e helum e cuniculi, small intestine epithelium, causing diarrhea and wasting. Also causes sinusitis, cholangitis and bronchiolitis. Conjunctiva and corneal epithelium, causing keratoconjunctivitis. Also causes sinusitis, respiratory tract disease and disseminated infection. Small intestine epithelium, causing diarrhea. Corneal and conjunctival epithelium, causing keratoconjunctivitis. Rarely, may cause hepatitis and renal infection. P. Stephora P. ronifia, skeletal muscle, causing myositis. Rapiera, 8 vesicularum, skeletal muscle, causing myositis. 8 canori, muscles, smooth and cardiac. Tracheoplestophora, T. hominis, cornea and conjunctival epithelium, leading to keratoconjunctivitis. Also causes myositis. T. anthropopteria, brain vitiforma v. corneal stroma, causing stromal keratitis. Nosema inocularum, corneal stroma, causing stromal keratitis. Microsporidium, M. silanensis, corneal stroma, causing stromal keratitis. M. africanum. Polar sac, plus, equals exospore, endospore, plasma membrane. Figure 1, Microsporidian spore. Box 1, acid-fast parasitic organisms, Microsporidia, spore, Cyclospora catanensis, oocyst, Lysospora B2, oocyst, Cryptosporidium pavum, oocyst. Common spores are 2-4 inches in size and oval to cylindrical in shape, with a polar filament or tubule. Figure 1, the spores are the infective stage of Microsporidia and the only stage of life cycle capable of existing outside the host cell. The polar tubule is an extrusion mechanism for injecting infective spore contents into the host cell. Spores are surrounded by thick double-layered cyst wall. Outer layer, exospore, is proteinaceous and electron-dense. Inner layer, endospore, is chitinous and electron-lucent. Spores are gram-positive and acid-fast. Box L, life cycle infection in host is probably by ingestion or inhalation of spores. In the duodenum, the spore with its nuclear material is injected through the polar tubule into the host cell. Enterocyte, inside the cell, the Microsporidia multiply by repeated binary fission, merogony, and produce large number of spores. Sporogony. Microspora. Box 2, parasites causing opportunistic infections in immunocompromised patients. Human immunodeficiency virus, HIV, positive cases. Common Microsporidia, Cyclospora catanensis, Lysospora B2, Cryptosporidium pavum, Toxoplasma gondii, Strongyloides stercoralis, Entamoeba histolytica. During sporogony, a thick spore wall is formed that provides environmental protection to the cyst. The spores are then liberated free from the host cell and infect other cells. Clinical features: they can cause wide range of opportunistic illness in patients with HIV and other immunocompromised diseases. Box 2, in patients with AIDS. Enterocitozoon benusi and encephalitozoon intestinalis lead to protracted and debilitating diarrhea in 10–40% of cases. E. intestinalis may also cause sinusitis, cholangitis and bronchiolitis. Infection with Plestophora can lead to myositis and E. helem can cause superficial keratoconjunctivitis, sinusitis, respiratory disease and disseminated infection. Stromal keratitis associated with trauma has been reported in infections with Nosema, vitiforma and Microsporidium in immunocompetent patients. Laboratory diagnosis. Microscopy. Diagnosis of microsporidiosis is made by demonstration of the spores in stool, urine, cerebrospinal fluid, CSF, or small intestine biopsy specimen. The spores can be stained with Graham's stain, periodic acid Schiff, PARS, stain, or modified trichrome stain. Note, spores of Microsporidia stain poorly with hematoxylin and eosin stain, although intracellular spores can be visualized by light microscopy. Electron microscopy is the gold standard. Identification of species and genera of Microsporidia is based on electron microscopy of spore morphology. Direct fluorescent method using monoclonal antibody is also used for detection of Microsporidia in clinical samples. Paniker's Textbook of Medical Parasitology. Cell culture. Microsporidia spores can be cultured in monkey and rabbit kidney cells and human fetal lung fibroblastic. Molecular diagnosis. Microsporidial deoxyribonucleic acid, DNA, can be amplified and detected by polymerase chain reaction, PCR. Common treatment. There is no specific and effective drug for Microsporidia. Intestinal Microsporidia may be treated with MEU, anidazole and albendazole, for superficial keratoconjunctivitis. Topical therapy with fumagillin suspension can be used. Prophylaxis. Improved personal hygiene and sanitation, especially in immunocompromised persons, can prevent Microsporidia. Key points of Microsporidia. Microsporidia are intracellular spore-forming protozoa, which belong to phylum Microspora. Spores of Microsporidia are oval or cylindrical in shape with polar filaments or tubules. Mode of infection: by ingestion or inhalation of spores. Reproduction. Microsporidia multiply by both merogony and sporogony. Clinical features: protracted and debilitating diarrhea and disseminated infection in eyes, muscles and lungs. Diagnosis: by demonstration of spores in stool, urine and CSF by Graham, S, PARS, or modified trichrome stains. Serological diagnosis includes direct fluorescent antibody test. PCR is also very useful. Electron microscopy is useful in species in identification of Microsporidia. Treatment: there is no specific and effective treatment. Intestinal Microsporidia can be treated with metronidazole and albendazole. Topical therapy with fumagillin suspension is used for superficial keratoconjunctivitis. Review questions. 1. Describe briefly the laboratory diagnosis of Microsporidia. 2. Write short note on the morphology of Microsporidia species. Multiple choice questions. 1. All are true about Microsporidia except A. First protozoan parasite studied by Louis Pasteur. B. Causative agent of silkworm disease. C. Extracellular spore-forming protozoa. D. Cause infection in immunocompromised subjects. 2. Laboratory diagnosis of Microsporidia can be done by all except A. Modified trichrome stain. B. Hematoxylin and eosin stain. C. Direct fluorescent antibody. D. Electron microscopy. 3. Enterocitozoon benusi preferentially infects A. brain. B. Conjunctiva. C. kidneys. D. Small intestine. 4. Microsporidial keratoconjunctivitis is commonly caused by A. Enterocitozoon benusi. B. Vitiforma. C. Encephalitozoon helum. D. Encephalitozoon intestinalis. Answer 1. C. 2. B. 3. D. 4. Capital C.